Let me get the thing here. Okay, so I'm going to adapt my talk a little bit today, and there's two reasons for that. One of them is John Gruber, who talked about a lot of the biologic clock data that we've been working on. And the other one is Max, who apparently was out so late last night that he doesn't want to hear his mentor give a talk. And he was talking about lipid aging clocks. And we're really excited about those things, but I'm not going to duplicate that, so we'll go in a little bit of a different direction today. I will say that our lab is extremely focused on both the interventional space and the biomarker space. And so increasingly, we have a lot of dry lab people in the lab working on generating biologic aging clocks and interpreting them and doing it in a meaningful way so we can get back to mechanism. So we're excited about that. And we're continuing to test a wide range of interventions across different species. I'll show you an example of one or maybe two. Ultimately, we're trying to combine these interventions together and figure out how to personalize them so we know who responds to what intervention at what time. And I think that, at least for the combination space, this is proving not as easy as we'd hoped. And so I'll suggest a little bit of the potential reasons maybe for that. And so I've used this slide a lot. I won't spend much time on it. There's no shortage of interventions out there. The things on the right are things that we have tested a lot. We're testing other things now. I should update the slide. Everything in green, though, we see some relatively between modest and really robust effects on health span. And so I think this is encouraging because many of these things came from work in other labs, and our ability to repeat results is pretty good. So I think that a lot of these things really are robust, and we need to test them in humans. I'm not going to talk about our human intervention studies today, but we are finishing an interventional study with alpha-ketoglutarate with Andre Meyer, and also well on the way to testing an interventional study with rapamycin in middle-aged individuals with biomarkers of aging. I think the challenge when we start the field right now, to me, is that we're almost a victim of our own success. We've had major discoveries in, I think, 3 of these different verticals that I'm pointing here: identifying interventions, diagnostics and clocks, and also understanding the pathways that govern aging. You could replace aging pathways with hallmarks. I completely agree with Roz that it really is a network, and so there's pluses and minuses to looking at things through the eyes of individual hallmarks of aging. But I think the challenge to the field right now is that we haven't figured out how to connect the dots between these things. And so, in other words, what we need is we need the data that says that an intervention, I chose AKG just for the sake of it, could be anything, which clocks they're impacting, which aging pathways they're impacting, what are the age-related disease outcomes that are most pertinent to that, and does it affect longevity? And this is not easily done because we can certainly test the interventions against the diagnostics, but if you're in humans at least, trying to get blood markers for all these different aging pathways is still a work in progress. So, but I think we need to focus on this because if we really are going to combine things effectively, we need to know which patterns are affected by which interventions, and that may begin to tell us how to put them together. So we're thinking a lot about this right now. I just want to make one other point before I go on. I'm also directing a center for ovarian aging, and there is increasing data that these geroprotectors extend fertility and delay ovarian aging. This is data from alpha-ketoglutarate, not from our lab, from China, but there's data on urolithin, on spermidine, on rapamycin, on metformin, and a wide range of other geroprotectors. And so what this may mean is that if you slow aging, you affect ovaries, or it may mean that part of the reason you slow aging is because you affect ovaries. And I think that we need to understand that better. So we're looking at a lot of geroprotectors in this context as well. I want to go on and say that there are a lot of things happening outside of small molecules, particularly in longevity clinics around the world, things from stem cells, exosomes, peptides, gene therapy, devices, other things. And I made this argument about 10 years ago with supplements because I felt like the aging field was really not engaging in doing the research on supplements enough. And so we really devoted ourselves to trying to do that. I think we need to do that now with these interventions. we need to figure out ways to work with these longevity clinics to really begin to understand whether taking a liquid nitrogen shower slows your aging or not. I'm a little skeptical, but we got to get the data and really begin to analyze it. So we're actively engaging with clinics now. And what this means is we used to have yeast and worms and flies and mice, but now we have billionaires as a new model organism for aging research. And this is, you might be skeptical, but actually billionaires are quite a lot like humans. And so I think we can learn a lot about humans from this. And I was talking to Carl Flager last night, and he made another point that's very valuable, which is that when we do mice, it's really hard to get aged animals to study. But if we're focusing on these billionaires, they're enriched in aged animals already. So I think that this is a really opportune time to take advantage of this data. And so I just want to show you one intervention, urolithin. And this is not something we developed. It came from Johan Oer's lab and other people in that research space. And this is a compound that can be made in the gut in some cases, some people. You get it from foods, from fruits and nuts. And like every other longevity intervention out there, there's data that it protects pretty much every organ system from everything. And so this is one of the things we really focused on, and we've been testing it. And this gives you kind of a flavor for how we go about doing things. And so we look across a wide range of species. In this case, we didn't look in worms, partly because it's been reported to have an effect in worms. There have been some data in mice as well, but we wanted to take it across a wide range of species. And you'll notice that killifish are in there. And I think killifish are still underrepresented for intervention testing. And we've been working on ways with our collaborators to deliver drugs in gels that the killifish eat. We've had, I think, a lot of success getting interventions to work there. So I would like to encourage other people to look at that. So we work with Nick Tolwinski in flies, and it extends lifespan in both males and female flies. And this is the data from killifish, and urolithin has a really robust effect on lifespan in killifish. You'll notice that rapamycin also does. That, to my knowledge, hasn't been reported yet in killifish, so we're interested in that. And one of the things about urolithin that we didn't expect, but we found, is that it reduces in muscle tissue mTOR signaling. And I want to come back to that in a minute. Here's the data in mice. So in mice, we start with 18 to 20-month-old animals. We treat them for about 8 months. And we use frailty indices, biomarkers of aging, and harvest the animals at 28 months. So we're not doing survival anymore in mice. It would be nice to have survival data. It takes a lot of time to do that. But I think also by harvesting the animals, we can take all the tissues and do all the mechanistic work in the mice and really begin to understand what the intervention is doing. If you let them die, you find them in a cage the next day, it's really hard to do that. So this is how we chose to do it. I don't think it's the only way to do studies. You can argue which strain, you can argue how to do it. But this is what we think is optimal for us. And so we see that if you look here, that this is 20 months, and so the frailty goes up in the control animals. It actually goes down at 2 different doses of urolithin at 2 months. And then it starts tracking back up with the control at the same slope. Now, at the low dose, you see that it goes up pretty rapidly at the end. I'm not sure, this is a one-off experiment. We're repeating it right now. I'm not sure this particular 2 points are that meaningful, but it's interesting. And it stays low at the high dose. One of the interesting things we found is that the effects are much more robust in males than females. We're also repeating that experiment because I want to confirm that, but that's preliminary data. And then we see a lot of evidence of improved muscle function, which is consistent with other reports. This is latency to fatigue on treadmills. It's improved by urolithin. And also the urolithin helps the mice maintain their weight later in life. Mice, it's true they gain weight throughout their life, but in the last 100 days or so, they become cachexic and start to lose weight. We see that delayed in the urolithin treatment. And so we've gone in and done a lot of omics. Look, this is transcriptomics, looking at the brain, and we do this using co-expression network modules. And this is just one result from that. I think that, again, it brought us back to translation and rapamycin and in this case, ribosomes. And so one thing we see in this case is that expression of this module of genes that's identified, basically it's the ribosome, goes up in the brain with age. Urolithin suppresses that. If we look across other interventions, AKG also suppresses this increase in ribosome expression with age. Rapamycin doesn't, but that may make sense because rapamycin is modulating mTOR activity, which is going to alter translation in a post-transcriptional way. So that's how we think about that. So again, we're back to the TOR pathway. So I am part of the TOR army, ROS, I guess. I keep trying to avoid it, but it keeps every intervention we look at keeps bringing us back there. And speaking of that, I wanted to tell you about a company that I talked about recently because it hits the same theme. This is called Q Biologics, and I'm now an advisor with them, full disclosure. And at first I was pretty skeptical of what they're doing, but I'm excited about it, and I think it turns out to be back to the mTOR pathway. So what they're using is inactivated bacteria that are injected subcutaneously to activate different organs, the innate immunity in different organs. And so generally how this works is that you do a sub-Q injection. It leads to cytokine release, activates innate immune system, which in part knows how to trigger the organ where that bacteria tends to infect. And so you get an innate immune response in the target organ, and then that can be used to treat a wide range of diseases. And so they are in stage 2 for various forms of cancer. In clinical trials, they're looking at IBS, they're looking at lung diseases. And so I think that when I looked at all of this data, what struck me is they're looking at aging, because they're targeting a lot of the inflammatory components of the aging pathway. And so they're also looking at metabolism and diabetes and obesity. And this is data from preclinical model done at McMaster University, where they improved glucose tolerance and insulin tolerance by using a intestinal targeted dead bacteria. And when I saw this, I'm like, I'm like a one-trick pony these days. I'm like, let's look at the TOR pathway. And so they had biopsy data from a human study, and this is transcription in the gut of mTOR in response to injection of this dead bacteria. So again, this is a relatively safe, robust way, I think, to turn down mTOR signaling if it's true. We're now going back and looking at the biopsies and really trying to dig into this pathway. So again, a completely different approach. It ends up in mTOR. And then, of course, I'm not going to talk about this today, but we've also shown that this drug, gemfibrozil, which affects frailty in female mice, it's supposed to be a PPAR alpha activator. It's not. Instead, it's blocking amino acid uptake or reducing it in the gut. And as a result of that, here's the reduction in amino acid uptake using ex vivo small intestines mice treated with gemfibrozil. I'm going through this fast because. But again, what that results in, less amino acids means less stimulation of the mTOR pathway. This is cell culture data. We have in vivo data as well on this now. And so these are screening for different interventions, talking to people that aren't even thinking about aging like Q, but now are thinking about aging. And I really think that regulation of mTOR is a central node that's targeting this aging process. And this may be one of the challenges to why it's hard to combine different interventions together. Because mTOR is triggered by aging, obesity, different diseases, and that leads to inflammation, and this is a cyclical process. And so turning mTOR back down may reduce this process and give you a relatively significant effect on aging, lifespan, and health span. But obviously, this pathway can't be completely turned off. And so if you start combining different things that hit mTOR from different ways, you might, instead of getting more benefit, start causing other problems that reduce the positive effect of the interventions. And so it's just the way we're thinking about it right now. I think if you have an intervention, it's worth considering whether it's impacting the mTOR pathway. I want to switch to worms, and this is a collaboration with Jan Gruber's lab and a grad student in my lab, Wei Han. Part of the reason I'm showing this project is I love this picture. This guy looks so gangster in this picture. I think when he submits his papers, I dare reviewers to reject them. It's even more funny if you know Wei Han. He's like the nicest person on earth. And so he's very interested in reanalyzing transcriptional patterns using worms, and Jan has accumulated a ton of data over the years. And so he's taking the approach of building gene expression networks as well with aging. So there's a lot of data you can model, define co-regulated genes as modules, look how they change with aging. You can also test it across a range of longevity interventions that there's data on, and identify these modules and test what they're doing with aging and what the interventions do. So this is the ribosome. I thought I would just keep on the ribosome today. This is a very tight module because all these proteins are extremely co-regulated. That's why I show it, because the module looks the best. But it's also relevant to what I'm going to tell you. And so there were 5 different modules that he was able to identify that are changing with aging in C. elegans. I'm just showing you one of them here to give you an idea how we're thinking. This is a mitochondrial module. And you can see at day 6, it has a certain level of expression. And then at day 8 and 11 and 14, it continues to go down. So expression of these genes are going down with aging. If you look at a short-lived mutant like DAF16, you get elevated expression. And then if you look across a range of longevity interventions from Jan's lab, the general trend is to turn these things down early in adulthood. So what that suggests is that these changes that are happening in this module are adaptive changes. The pathway is going down, and that's helping the worm stay alive longer. And if you turn it down prematurely, you pre-adapt the worms to that change, and the worms live longer. And so these are the 5 different modules, and you can then go back and do go back to human cell culture data and look at a wide range of drugs against orthologous modules. And so 4 of the modules go down with age, like the mitochondria, that's these 4. One of them goes up with age, that's the ribosome. And then you can say, where are the longevity extending drugs? What are they doing in cell culture? And a wide range of them are in this adaptive mode. So they turn down these pathways. And so presumably what's happening in the worms is they're turning down the pathway early, and that's good for aging. Now you'll note that this is mTOR inhibitors and a wide range of other things. You'll note that they also turn down the ribosome pathway. And so if you infer back to worms from this, what it might mean is that the ribosomal upregulation with aging is a maladaptive response, and turning it down may be beneficial for aging. So the hypothesis is these 4 changes are adaptive changes in the worms, and this change is a maladaptive change. And then you have an enrichment for longevity genes and things that regulate these pathways. Now, I'm curious also about what the drugs are up here, and so we're going to go back and test them, because it may be that altering the pathways back up could have a benefit in other contexts with aging. So I think this is a new way to look at how drugs are impacting the aging process. So we just chose 10 at random. We took criteria that are even semi-defensible to start with. Here's the effects of 10 different genes, and then 3 controls. Rapamycin's a control, so it fits into this adaptive module. Aspirin and curcumin don't. They seem to be doing something else, and so that's what I mean by saying that there may be other drugs out there that are doing something else to impact aging besides this adaptation. And then we just tested these 10 drugs. We focused on things that were not in known drug pathways. And 7 of them, I won't go through the data, but 7 of them extend lifespan. So we're highly enriched now in finding new drugs that are not in old classes that are impacting aging in worms. And so I think this goes back to Matt Kaberlein's point that we've really only explored a very small component of the drug space in terms of what could affect aging. And maybe if we really want to combine things together in an effective way, we need to explore a much broader space and find other classes of drugs that we don't even know about yet that are impacting aging. And that's what Weihan's trying to do. And that's a lead-in to Aura Biomedical, which is mainly coming from Matt Kaberlein's lab. I'm also a founder of this company, just to disclose. But it's largely people that came out of Matt's lab that created this worm bot for high-throughput worm screening. And so this is ongoing now. Thousands and thousands of new drugs are being screened. And they thought about doing the million molecule challenge. We're not quite at 1 million yet, but this is an interactive challenge, and so that's why I want to bring this up. You can support a small amount of money. It's like $100. Pick your favorite intervention, have them test it, and they will publish the data and put a leaderboard of interventions that affect aging in worms. So I think this is relevant because you may be working in another organism like mice or even humans with a particular intervention, and you may or may not believe that worms can tell you a whole lot, but you can get really high quality data on any intervention you care about just by doing this, and then you have the rights to do whatever you want with the data. So I encourage other people to do this. It gives them data at a relatively at a very cheap cost and a very high quality data. You'll see the leaderboard right now. There's some rapamycin stuff in here, berberine. Sildenafil is interesting. I'm not sure that really is going to impact aging, because if it really impacted aging, we'd have a lot of middle-aged men that were looking a lot younger right now. But it's an interesting molecule, and a lot of other things are being proposed. And so this may be a way of finding new ideas as well. So I'm actually talking fast today, so I'm only have a couple slides left. So I think what I want to do is leave you with a thought. And this comes back to what I was saying earlier, that a lot of these interventions are having modest effects on aging, at least in animal models. I think they will in humans too. And so it's important to test these interventions preclinically, clinically, and then to get into community-based studies if you can, to validate that we're really modifying the aging process. Even if it's a small effect, I think this will be a revolution for the field if we can really nail it down, because it will increase awareness and excitement, demonstrate that health span can be affected, and cause an even higher avalanche of resources to pour into the field. But I think there's another point that we don't want to forget, and that's that a lot of us, including I think Aubrey would vote for this, we really want to have dramatic effects on health span and lifespan. I think you might argue whether that's a good or bad thing, but it's certainly one of the most important questions in biology. Can we dramatically impact the aging process? Is it possible to stop aging? Is it possible to reverse aging? I don't think we know the answer to that right now anywhere. I'm hesitant a little bit about the field right now, because so much is being poured into translation and moving forward with the interventions we already know. But I'm skeptical that any of those interventions are really going to take us from here to here. And so we still need to focus on basic discovery science and maybe be thinking about interventions in an entirely new way if we're going to, start to talk about tenfold effects on aging. And so I think that we still need to encourage government agencies, nonprofit funding agencies, and anybody else who wants to support this basic science, especially crazy ideas to understand aging. I would point to Peter Fedichev thinking about, this is more of an entropic problem, and if you think about aging as an entropic problem, it would lead you to a completely different kind of set of interventions that generally haven't been tested yet. So just a word, we need to keep funding the early part of the pipeline too here. Actually, maybe just the liquid nitrogen showers will take us from here to here, I don't know. So with that, I just want to say, I think most of the people in this room know we now have 115 webinars on YouTube that are all free. You can register to watch these shows live, or you can go find them all and watch them whenever you want. So anybody that's interested in seeing more one-hour discussions on every aspect of aging that you can think of, please go there and watch the videos. And with that, I have a lot of collaborators. Andrea Meyer is really a partner in crime at NUS, and a lot of people that really helped on these projects. And I left us time for questions, Aubrey, and catching you up on time. So thanks.
Yeah, thank you so much, Brian. What questions do we have for the wonderful Brian Kennedy? Okay, where's the microphone?
Wait, this is hard, isn't it?
David? There you go. Whichever.
Thanks. Brian, I wanted to ask you about this strange species that recently being tested more often, the billionaires.
Yeah.
To what extent can we get more billionaires recruited into this test?
Yeah, well, maybe if I stop insulting him, it would help. But I think that I'm actually very, supportive of this. I think we have to keep focused on democratizing, health span. At the end of the day, we have to all be aware that we're really out there for the billions of people that are getting old, not just the billionaires. But these clinics are generally focusing on high net worth individuals. They're charging somewhere between $5,000 and $100 or $150,000 a year to tell people they're going to optimize their health and extend their health span. And I think it's a shame if we're not capturing that data in some way and analyzing it in a high-level statistical way. Most of these clinics are not, really doing AI-based modeling of their data and trying to understand what's working. These things are being tested. Many of them I can't really do academically. I can't do a large-scale stem cell study very easily, measuring aging markers over a long period of time. These things are happening in clinics, and I think we need to figure out a way to work with them that's effective. Now, it's going to require flexibility because clinics, are protective of their data. They're not going to be excited about negative results, and so it's not an easy problem to solve. But I think that as academicians, we have to engage and really try to maximize the value that comes out of these studies.
Yeah.
Hi.
Thanks a lot, Brian. Regarding dramatic effects, could drug combinations or combination in general be a solution by looking for drug synergy? This is very heterogeneous. You can even have androgenicity effects. But do you know any drug synergy cases where you had a dramatic increase?
I think they're few and far between right now. If you look at longevity, there is metformin and rapamycin, but it's a very small additive effect. And those are the kinds of things that we've seen. We do see that periodically, but we're not seeing like massive magnitude changes. And I know Aubrey's testing this too. And I'm still confident that we can find those, but I just think it's not going to be trivial. And so when you have somebody out there promoting taking 12 different things at once, it's like throwing 12 colors of paint together and you get some ugly form of gray. I really don't know what's happening. And so anybody that wants to do that, I think they should be doing testing of that particular combination in people and getting actual data on it, because I can't predict what's going to happen. So.
Yeah, our hypothesis is that we're going to have somewhat more predictable and perhaps somewhat more high magnitude synergy when the things that we're combining include gene and cell therapies and not only pharmacological ones. But I guess it remains to be seen. A colleague.
Hi, Brian. Thanks for the great talk. Quick question. So the newest ITP paper is out, and AKG didn't do that well. So any thoughts on that?
Well, we didn't propose AKG, and the reason is if you look at the mouse data on AKG, the extension of lifespan is only around 5%. The robustness of the effect is in health span and frailty. And the ITP, I've supported them for many, many years, but I feel like they haven't really still embraced this concept of measuring frailty and health span in the animals. The other thing is that they didn't use the calcium AKG. They used a straight version of AKG, which we have seen lesser effects with, and we abandoned that in the clinical approach because it's acidic and it can cause like transient heartburn when you take it. So, it could be that it doesn't work in the genetic outcross model, but we need to really dig into the frailty and the health span parameters, because that's where we reported the benefit in the first place. So.
All right, thanks, Brian.
Thanks.